afternoon, everybody. My name is Georgina Mawer. I'm Chemical and Regulatory Specialist at BLC Leather Technology Centre. I'm joined today by my colleague McKenzie, a research scientist at BLC. We would like to talk to you today about a joint project between BLC and Applied DNA Sciences, ADNAS. This is DNA labelling, the smart way to trace materials through the supply chain. Given the emerging global reality of sustained volatility and rapid change, the need for robust product traceability and transparency is even more critical. However, many global supply networks are poorly equipped to respond to such challenges. Moreover, the significant threat posed by environmental and social risks to brand value ensures that such challenges are increasing. Failure to adequately address supply chain challenges could leave companies exposed to undue risk and scrutiny, which may include trade issues or associated prosecution. The leather making process potentially encompasses a myriad of environmentally and socially sensitive aspects, including animal welfare, labour compliance, and environmental pollution. It is also important to note the increasing scope of NGO activity across the leather sector. The industry continues to experience increasing pressure, notably in relation to deforestation, effluent discharge, use of hazardous chemicals, and animal husbandry. Inevitably, this critical examination of our supply chain will grow, as will the NGO's influence on government policy and public opinion. A concerning development is the adversarial approach of certain NGOs in response to perceived failure or inertia on parts of the brands and retailers in enacting change. Lastly, the leather industry is subject to a suite of national and international regulatory controls, specifically concerning upstream processes. Failure to comply with such controls can manifest in dangerous or defective products, as well as detrimental effects to the production process itself, thus presenting a considerable risk. The leather industry supply chain is truly globalised. Typically, the supply networks of leading brands and retailers comprise multiple tiers across numerous continents. Accordingly, the complexity of such supply chains directly contributes to difficulties in management and is further exacerbated by the vast networks of independent suppliers, manufacturers, and raw material sources across the leather sector. Significantly, BLC studies suggest that a typical footwear supply chain could incorporate hundreds of different tanneries. In turn, these tanneries could be sourcing from as many as 500 slaughterhouses and potentially millions of farms. The next few slides serve to represent just that. Tier 2, 20 Category D tanners. Tier 3, 120 Category A tanners. Tier 4, 480 slaughterhouses. Tier 5, 1.2 million farms, and 24 million cattle. It is these combined factors that serve to emphasise the strategic importance of thorough supply chain management processes. Consequently, traceability is absolutely fundamental in identifying and understanding networks of supply, and ultimately will deliver robust oversight and management of supply chain challenges. As highlighted earlier, many companies have very little understanding of where their leather comes from. Perhaps DNA could be the answer.
Thank you. So, could DNA be a solution? DNA can be considered as nature's ultimate means of authentication for supply chain protection and form of evidence for crime prevention and security. But what exactly is DNA? DNA stands for deoxyribonucleic acid, and is a genetic material located in the nuclei of cells, which comprises the genetic blueprint of an organism. DNA can be considered as the building block of life and was first discovered by James Watson and Francis Crick in the 1950s, although more recent publications try to dispute this. DNA is structured in the form of a long polymer, double helix structure consisting of 4 base pairs, which are also referred to as nucleotides. These include adenine, A, cytosine, C, guanine, G, and thymine, T. The bases of one DNA strand complementary pair with the bases on the other, always binding adenine to thymine and cytosine to guanine via hydrogen bonding. The order in which these bases pair forms the genetic sequence comprising the instructions of the genome. Genes are short sections of DNA, defined as sequences of nucleotides forming part of the chromosome which code for a particular protein. DNA can be considered as a dynamic and relatively robust polymer in terms of its ability to survive harsh conditions and prolonged periods of time. For example, the oldest human DNA discovered dates back to over 430,000 years, and the oldest botanical DNA dates back to potentially over 800,000 years ago. With this in mind, the concept of applying a DNA marker to level manufacture with relatively harsh conditions in terms of pH and temperature holds great potential. Applied DNA sciences are the creators of a successful and patented product known as signature DNA. Signature DNA forms the core of the precision-engineered markers that ADNAS use for various forensic applications, including supply chain traceability. Referred to on this slide is signature tDNA, which refers to the DNA markers created for the application to textiles. The concept, innovation and proven success of this product is what we would look to apply to the leather industry also. Essentially, signature DNA, as the name suggests, is a unique DNA marker created by taking long strands of botanical DNA and segmenting them into smaller segments. These segments are then encrypted, reshuffled, and reassembled to form a unique, secure signature DNA marker. Additional layers of protection and complexity are applied to prevent these markers from being decoded by conventional techniques. Signature DNA can be engineered with infinite variability and can be individually customized and embedded into a range of host carriers, including inks, varnishes, laminates, metal coatings, and the list goes on. Having discussed the concept of DNA markers, it is important to address the misconceptions and negative connotations that are often associated when discussing DNA and genetic material, particularly with regard to genetically modified organisms. The World Health Organization has defined genes as a length of DNA that determines the order of an amino acid to make a protein. The definition of genetic modification is the alteration of genetic material via genetic engineering techniques. The ADNAS markers have been designed so that they do not have sufficient length to make proteins and lack any function. The marks are in no way similar to a gene and can therefore be disregarded as being genetically modified. Signature DNA is chemically identical to the DNA found naturally in food and therefore presents no risk to humans who may ingest it, even at the extremely low levels that we use. ADNAS's DNA markers are considered to be generally recognised as safe, based upon scientific procedures and on their chemical identity to molecules that we have consumed for many years as part of the human diet. On the topic of queries in genetic modification, we wanted to share with you a short video. This features Dr. Mike Hogan, who is based at ADNAS Stony Brook in New York and is one of the principal DNA scientists working on this project.
From other kinds of molecular marks. The most important thing to recognize is that it's made out of the 4 natural building blocks, A, T, C, and G. And because it's really a natural product, it's something you literally ate a gram of yesterday. It's intrinsically safe. It's also a polyester, which allows us to formulate it into different products. It's kind of a natural polymeric material. And it's an important thing to recognize is as such, it's not human, it's not pathogenic, it's intrinsically safe. In terms of myself, I have a background in biology from Dartmouth and Yale, Stanford. I was an academic for about 20 years at Princeton and then Baylor College of Medicine. I've been involved in about 5 startups. My job is to help bring that expertise to the development of the pharmaceutical and the biotech aspects of what we do here. I'm VP of Life Sciences at Applied DNA, and I oversee much of the research in pharma, textiles, and military applications. It's really important to know why applied DNA is applicable and safe for pharma. It's not human. It's very small. It's originally derived from plants. We'll talk about that in a little bit. And it's very short. And because it's short, it couldn't possibly have any genetic function. It's really just a small bit of natural product. It started out life being from plant origin. They've been reshuffled to generate codes. We make these A, T, C, and G codes in a way that they're kind of like a natural molecular barcode. If it were a barcode, it'd be as long as your arm. And because they're made out of DNA, we can use a lot of the same tools that were derived from forensics to really analyze them. In terms of the FDA, it's important to recognize that they already have given a lot of thought to the safety of DNA. They've already written white papers in 2010 that say that DNA is safe at up to 150 micrograms of DNA per dose, which is enormous. It's about 1 billion times more DNA than we would put into any formulation. And they've already recognized that physical chemical identifiers are allowable in pharmaceutics, in fact very important, and then molecules like DNA fall into that category. To sum up, it's important to recognize that DNA is very safe. It can be introduced into pharmaceutical doses at 1 billion times below what's allowable. The FDA has already given a lot of thought as to how much DNA and what kind of DNA can be introduced into any dose. The bottom line being that our DNA is so short that it's intrinsically safe and 1 billion times less than what the FDA allowed. It's also important from a practical perspective to recognize that our DNA is almost completely incopyable, so that if a pharmaceutical company were to introduce it into its formulation, it couldn't be challenged, it couldn't be counterfeited by others, and really importantly, because of the way it's made, it's naturally suited to be analyzed by the very same techniques that have been developed in forensics and food safety, so that it can be routinely measured not only in a centralized lab as we have in Stony Brook, but even out in the field.
I'd like to take this opportunity to talk you through a couple of ADNES's success stories with DNA labelling. The first case study, how signature T DNA in finishing treatment assures authenticity of finished fabrics. This particular project was conducted for a textile solution provider based in the US, who provides performance chemistry in treatments for fabrics. Their products are heavily relied upon and trusted by top designer brands, and many of these products are marketed and sold to the home textile and apparel industries. What they were looking for in ADNES was to confirm a finishing treatment is being appropriately applied to fabric and to authenticate said fabric treatment. ADNES trialled the addition of signature T DNA through a program of testing. They collected 12 samples, 4 of which act as negative controls, the other 8 having had the DNA labelling. All 12 samples were submitted and analysed for a unique signature T DNA profile. In addition to this, 16 samples were analysed to ascertain whether they had been adulterated. There was a successful recovery of the DNA profile from all 8 positive samples, and no recovery in the 4 negative controls. In addition, all 16 samples were confirmed as adulterated in some way. From this project, we learned that signature T DNA can be applied in the final processing stages, like finishing treatments, to ensure durability and maximum performance of finished goods, as well as to detect the application of adulterated treatments. The second case study I want to share with you today, and perhaps ADNES's biggest success story to date, is with regards to the cotton supply chain. Consumers rely on brands they trust to ensure that the products they buy are what they say they are. A good example, and common misperception in today's textile world, is that Egyptian cotton is the best. Consumers buying sheets made of Egyptian cotton expect a luxurious night's sleep because of the association with presumed premium quality. Government agencies like the FTC establish guidelines to protect the consumer by ensuring that products are based on accurate information on product origin, construction, quality and care. The disclosure of fibre content is integral to trade in textiles and apparel. It is incumbent on the brand and its supply chain partners to know and correctly report fibre content in both documentation and on product labels. If a product is labelled to use a single fibre, such as 100% Egyptian extra long staple, that's ELS for short, then there is no tolerance for the use of any other fibre type. In 2012, global cotton prices rose to record levels, especially for premium ELS cottons. Historically, the goal of select processors of premium cotton fibres, such as Egyptian ELS or American Pima, was to buy the highest quality cotton at the lowest price, attempting to meet product specifications by blending bales with different fibre qualities and types. The blending of premium ELS cotton with shorter, non-ELS upland cotton occurs when manufacturers search for higher profit margins, or if there is a shortage of available high-quality ELS fibre. This blending of different fibre types results in impure products being labelled as 100% ELS, violating US and international laws. Within the home textile industry, retailers and brands have relied on documentation and other forms of inspection that provide no clear confirmation as to the fibre content of finished label product. Working with cotton trade associations, ADNAS validated and commercialised the methodology to identify the genetic variations between Pima cotton and upland cotton. This proprietary technology, which is setting new industry standards, is known as fibre typing. Fibre typing offers a diagnostic DNA test that can help verify fibre content and assure quality and labelling compliance. It provides a forensic method of authentication for Pima cotton products, but it does not offer country of origin at this time. Cotton fibres start to mature around 45 to 60 days post-anthesis, or DPA, and fibres start dying sometime after day 40. When a cotton cell dies, the DNA becomes fragmented and degraded, making it far more difficult to analyse by the polymerase chain reaction. Nuclear DNA is nearly completely degraded after 45 DPA, making any claims for nuclear DNA analysis using mature cotton fibres very difficult. In 2009, ADNAS discovered the survival of the chloroplast DNA in mature cotton fibres for its fibre typing DNA assay. ADNAS has a solution that uses forensic science to tag, type, and test textile products. The core technology of fibre typing has a unique molecular tag that marks cotton fibres. Fibre typing can detect as little as 1% upland cotton in Pima-labelled textiles. In 2009, ADNAS conducted a market sweep of 100% ELS or 100% Pima-labelled bed linen. 29 samples were selected at random from multiple online sites and retail outlets. The fibre typing results indicated that a non-compliance occurred in almost every sample. 83% of the products tested did not contain 100% Pima as was promised on the label. In other words, only 17% of the samples tested actually complied with the information provided on the product label. Since its initial testing, ADNAS have processed thousands of tests for many brands, manufacturers, and other cotton supply chain partners. We are now at the stage where over £100 million of cotton has been tagged. The DNA, which is applied during aerosol during the dehydration step, only needs to be added in 1 part per trillion of cotton. The bottom line for all of this is that consumers will buy products from sources that they trust. Retailers and brands will buy from suppliers that provide truthful materials. To do this, suppliers will need tools that provide them with the certainty that the materials they use are really what they say they are. DNA technology has proved to be that solution. I'll now pass back over to Diva, who will discuss how DNA labelling can be applied to the leather supply chain.
So, why do we want to protect leather? Traceability refers to the completeness of information in every step in the process chain. The need for traceability is ever increasing in a wide range of industries, and the leather industry is no exception. This need is mainly driven by consumer pressure with regards to having insight and knowledge of where products are sourced. One of the key areas where traceability could be considered a massive advantage in the leather industry is in the trade of split leathers. Split leathers are sold onto traders, and often traders combine splits from multiple supplying sources. Therefore, with insufficient traceability processes in place and limited options available, more often than not, the original source of the split leather is lost forever. DNA technology poses a potential solution for the protection of upstream sustainability in the trading of split leathers. Additionally, having transparency and traceability throughout the supply chain enables ethical and environmental claims to be verified with regards to issues such as sustainably produced, no deforestation, responsible chemistry, and modern slavery. BLC and ABNAS have developed a DNA marker application flowchart that we hope we can develop to manage supply chain complexity by using DNA to bridge the gaps in the supply chain. Theoretically, each supplier will have their own unique DNA marker that they apply at the applicable stage, addressing the key points in the leather manufacture process, which we have acknowledged as being the farm, the slaughterhouse, the wet blue tannery, the finishing tannery, and eventually the manufacture or brand. We propose to apply DNA at the following points. Firstly, at the farm to the live animal. We propose to apply a short-term marker similar to the spray paint markers already applied to cattle as the animal leaves the farm immediately prior to slaughter. This will be in a single location and will subsequently be authenticated at the slaughterhouse. Secondly, the slaughtering facility. At the slaughterhouse, the authentication of the farm-applied marker will be conducted, and an additional marker can theoretically be applied. This marker would be similar to the one applied at the farm and would be applied in a single location via spray application. Next comes the wet blue tannery. At the wet blue tannery, authentication of the slaughtering facility marker and potentially the farm marker, if it can be modified to sustain the length of time, could occur. It is likely that during the tanning process, the markers applied at the farm and slaughterhouse will be destroyed, although this is something that we need to investigate. The markers, therefore, the wet blue marker tannery will be applied during the actual tanning process. The markers therefore need to be able to withstand the conditions of tanning, including chemicals, fluctuating pH and temperature, but they also have to have a high affinity for collagen in order to bind throughout the entirety of the hide and penetrate into the full thickness of the substrate. At the finishing tannery, authentication of the wet blue tannery marker will be conducted, which will aid in tracing back to the original source of wet blue, which will be beneficial for the trading of splits. Additionally, a final DNA marker with a unique finishing tannery code could be applied and incorporated potentially during wet processing in the production of pressed leather or in the finishing application themselves, potentially as an intermediate or impregnation code. The authentication process can only currently be conducted by ABNAS at one of their laboratories. However, an in-field test has been developed, but it is not commercially available as of yet. The process of authentication involves polymerase chain reaction and capillary electrophoresis for marker identification. An additional point that I would like to clarify at this point is that it is important not to confuse authenticating DNA markers as being the same as genotyping. Genotyping in leather is not a novel concept and can be used to aid in species identification. However, when authenticating, we are not examining the animal's DNA. Instead, we are simply looking to identify a marker that we have knowingly applied. So, the project. The project is a BLC and ABNAS-run research programme. We have 5 major brands as partners and sponsors of the project. From recent press releases, we are able to disclose that Clark and Puma are sponsors, but the remaining sponsors would like to remain anonymous at this stage. Although the project has just gone live, we are still seeking additional partners that may wish to be involved. There is a limited window of opportunity in which still to join, but if anyone finds interest and would like to be part of the project, please let either Georgina or myself know, and we can facilitate with any questions that you may have. Currently, we are in the early stages of the project and we are assessing the feasibility and the proof of concept. BLC is supporting ABNAS with details of leather manufacture, and ADNAS are attempting to modify DNA markers accordingly for each of the different application points. Small-scale experiments and authentication will be trialled, and depending upon the results, we will look to scale up the project and run larger-scale trials where possible. The project intends to run for 6 to 9 months, and for simplicity at this stage, we will focus on a standard chromium tanning shoe upper manufacturing process only. However, towards the end of the project, we look to engage the supply chain fully and to fully assess the feasibility on an industrial scale. The next couple of slides are just references to some recent press releases that have surrounded the project, particularly after Hong Kong. We've had a few press releases. Georgina and I would like to thank you for your time today. We hope that you find the concept of DNA traceability as interesting as we do, and we welcome any questions